The use of AAA ATPases to remove proteins from a lipid bilayer is a common theme in protein quality control. A mechanistic understanding of this essential process requires a reconstituted system. Previous reconstitutions with AAA ATPases were complex and heterogeneous. Our system is simple and fully defined. This allows us to manipulate the AAA ATPase Msp1, the substrate, and the lipid environment.
To begin, add previously prepared reconstitution buffer, purified Msp1 and TA proteins, and liposomes in a PCR tube, then incubate the mixture on ice for 10 minutes. During the incubation, cut the tip of a P200 pipette tip to about 1/8 of an inch in diameter. Next, vortex the tube containing biobeads thoroughly to obtain a uniform mixture. Then, quickly remove the lid and use the cut P200 pipette tip to transfer an appropriate volume of the beads to an empty PCR tube. Once the 10-minute incubation for reconstitution is complete, using an uncut pipette tip, remove all liquid from the biobeads. Then, transfer 100 microliters of the reconstitution into the tube with the biobeads and allow it to rotate on a wheel for 16 hours. The following day, spin the tube in a picofuge to pellet the beads, then transfer the reconstituted material to a clean PCR tube and keep the tube on ice. For removing proteins that failed to reconstitute into the liposomes, equilibrate the glutathione spin columns with extraction buffer, which typically involves 3 rounds of washing with 400 microliters of buffer, followed by centrifugation to remove the buffer.